Good afternoon. I'm Jim Morrison, the president of the Hudson River Environmental Society. Welcome to part 3 of our seminar series entitled The Drivers of Change, Climate, and Introduced Species. We anticipate you'll find today's presentation both informative and enjoyable. 51 years after being established as a non-advocacy organization, one of HRES's ongoing missions is to provide a platform through our symposiums for open discussions and sharing of data between the research communities, academia, resource agency scientists, political decision makers, and of course the citizens of the Hudson Valley. Please check out our website at hres.org if you'd like to learn more about us. It's now my pleasure to introduce Mr. Clay Hiles, the Executive Director of the Hudson River Foundation.
Thanks, Jim. With the modesty of a lesser organizer of this program, let me say to all of you, especially to those who are participating today for the first time, that this is turning out to be really one of the ironically wonderful consequences of the terrible strangeness through which we have been living for a year now. The annual conference of the Hudson River Environmental Society has always been an important moment in the rhythm of environmental interests in the Hudson River, both for the substance of its presentations and discussions and the opportunity for our community to gather together. But this year, with the leadership of the Society and its president, and his and our energetic and irrepressibly determined colleague, Chuck Nieder, the heretofore one-day event has become a 5-part exploration of what we have always wanted and needed to know about fish in the Hudson. And from the vantage point of the Hudson River Foundation, what we want and need to find out going forward. HRES has generously allowed the foundation to collaborate in this program in a way that will guide our efforts to shape research priorities for science, including social science, for the Hudson River and watershed for years, even decades to come, and to form the backbone of a comprehensive new strategic plan for the foundation being developed under the expert direction of the foundation's about to be executive director, Jonathan Kramer. So with thanks and kudos all around, let's begin with Jim Lodge's explication of the mechanics of today's session, including the ways in which you can get all your questions about everything fishy answered. Jim?
Thanks, Clay. Good afternoon, all. I'm Jim Lodge, Hudson River Foundation. So this seminar is being recorded, and we're intending to keep the talks right at 20 minutes. So and have all questions and answers at the end of the 5 sessions, 5 speakers. So we encourage you to use the Q&A box to write in your questions, and to help us keep organized, we request that if you have a specific question for a speaker, just put their name first and then what the question is, and then Chuck and I will gather them all, and again, we'll answer them at the end. In the unlikely event that there is a technical problem, we will stop this presentation and email all a new link probably about 5 minutes after that happens. Again, just in that unlucky event of a major technical problem. If you are experiencing other technical issues, just please also write them in the chat and we'll work to resolve them. Chuck.
Thanks, Jim. So planning for this started long before pandemic disrupted our lives. So we've actually been planning this seminar for a couple of years. By the time we got around to figuring out how we were going to run this this year, virtual conferences and seminars were becoming a norm, and so we went ahead with that platform to offer this series. So the content, it's a 5-week series, 2 hours a week. Each session is specific, has its own theme. So in each one alone tells a story, but collectively they tell a bigger picture here, and hopefully give you a better understanding of the Hudson River estuary and its environs. And the other unique thing this year, we really did team up quite closely with Hudson River Foundation. A few reasons. One is they have close ties with the research community. The second reason, as Clay said, they're looking at developing their next strategic plan to figure out where they're going to fund research in the future. And finally, this seminar series is dedicated to Dennis Siskowski, who is a true friend, mentor, and colleague for many of us. And so I just want to thank you all for participating today. And I want to thank the speakers for taking the time to put together such great talks and agreeing to pass this information on to you. So without further delay, I'll introduce our first speaker, Dr. Catherine Mills. So Dr. Mills studied environmental science and policy at Duke University, and then went on to earn her master's and PhD in natural resources from Cornell University. She currently is a research scientist at the Gulf of Maine Research Institute in Portland, Maine. Her research interests are to look at how physical and ecosystem conditions are changing, how these changes affect fish populations, biological communities, and marine fishes, and how fisheries and fishing communities can adapt to these changes. Dr. Mills is an awardee of a Pew Marine Fellow, and she currently leads a global working group on climate resilient fisheries. A little more background on Dr. Mills. She spent quite a bit of time in the Hudson Valley when she was working on her master's. She was working with Mark Bain. She spent many days in a canoe in the marshes, tidal marshes along the Hudson River, where she studied fish communities and estuarine wetlands. So she's pleased, and we're pleased to have her back in the Hudson Valley, so to speak, at least virtually, to participate in today's symposium. So thank you very much, Kathy, and take it away.
All right, thank you very much, Chuck. Let me see if I can get my slides up. Okay, are the slides showing? I can't see anyone, so.
Yeah.
Okay, great, thank you. All right, well, thank you all for joining today. I am here to present a bit of an overview of ocean warming. So looking a bit offshore for the bulk of my talk, to think about ocean warming, its effects on fish in the Northeast US. Many of these species rely on the estuary at some portion of their life history. So trying to start understanding some of the change that's happening in the ocean and how that might be affecting fish populations that intersect with and interact with our estuarine environments as well. So in the talk, I'll be starting with an overview of warming on the Northeast US shelf. And when I talk about the Northeast shelf, what I'm really talking about is the area spanning from the Gulf of Maine, including Georgia's Bank, down to Cape Hatteras and the Mid-Atlantic bights here in the center of those areas. I'm going to be providing an overview of 3 different ways in which warming is, how we see climate affecting temperature trends and patterns on the Northeast shelf. So these come through in our warming trends, as well as the timing of warming and cooling and temperature events. Then I'll be connecting some of these changes in temperature to species, particularly fish and marine invertebrates, through shifts in their distribution, population productivity, and phenology, or the timing of life history events. And I'll wrap up. I want to just acknowledge that my talk is very temperature focused, so I'll conclude with a few insights into other climate-driven changes that we may want to keep on our radar screen and consider as well. So the Northeast shelf has been experiencing rapid warming in sea surface temperature since the satellite sea surface temperature record really began. So the satellite record goes back to 1982, and from 1982 through 2020, the Northeast shelf has been warming about 0.03 degrees Celsius per year. The global average is about 0.01. So you can see that over a long span of time, the Northeast shelf has been warming about 3 times faster than the global average. But what's particularly exceptional is that in the more recent years, so here I've pulled out the last 15 years, which is ecologically meaningful for a number of species when you think about their lifespans. During that time period, sea surface temperature on the Northeast shelf has warmed at about 0.06 degrees Celsius per year. So about double the long-term warming rate and about 5 times faster than the global average warming rate. So this region has been experiencing exceptional warming. To put this another way, what you see here in yellow are the pixels around the globe where the warming rate has been faster than 95% of the remaining pixels in the globe. So if it shows up in yellow, it's warming faster than 95% of the world's oceans. And you can see that the whole Northeast Shelf has really been experiencing quite rapid warming rates relative to the rest of the world. And our climate models project that this rapid warming in this region relative to other regions will continue into the future. So you can see it's also popping out in bright yellow in future projections, going out in this case to around 2080 as well. So the ocean has been warming really rapidly, but thinking about a number of species that use estuaries and that migrate through habitats, it's also really important to think about how consistent warming is across different habitat types. So I'm sharing this slide with you today, but this is really some fairly preliminary work that we are trying to pull together right now. And what we're looking at here is for the Hudson, what warming rates look like as you go from the ocean, which is at the bottom actually, through the coastal waters into the estuary and then into the river itself. And what we would hope to see is that these align fairly well so that habitats are changing at similar rates. And these figures here show that in some seasons that is the case. In fall, for example, these all line up quite tightly, but in other seasons like winter, the ocean is warming much more rapidly than coastal waters, which are warming more rapidly than the estuary and rivers. So this poses potential implications for migratory fish and particularly anadromous and diadromous fish. So this is something that we'll hear a bit more about in the next talk, thinking about how warming and phenology of fish migration and spawning are all interconnected. So another temperature-related change that we're seeing is changes in the timing of warming and cooling, or essentially the seasonality of temperature across the region. I've pulled out here just one figure from some work that Andy Thomas led a couple of years ago, looking at what we see in terms of the annual cycles of temperatures when ocean waters warm up and when they cool down. So Andy set temperature thresholds based on the long-term warming patterns and defined when we saw the start of summer defined against a temperature threshold and when we saw summer end each year. And what we see from this work is that the length of the summer-like period is increasing substantially all along the Northeast Shelf, particularly in the Gulf of Maine, where the summer-like period has been expanding about 2 days per year since 1982, when our temperature record began. But it's also expanding about half a day per year in the mid-Atlantic. And this is also really interesting because in the mid-Atlantic, the expansion is more related to summer starting earlier, so the spring warm-up occurring sooner, whereas in the Gulf of Maine, it's more related to a longer fall or longer extension of summer into the fall, so the cool down coming later in the year. And then finally, just a third aspect of temperature change that we're experiencing in the region relates to marine heat waves. So we define heat waves as a period of time that lasts more than 5 consecutive days, where temperatures are at or above the 90th percentile of past temperatures that have occurred on that day. So we look at the climatological temperature record and then pull out periods of time where temperatures exceed this certain 90 percentile threshold and last for a fairly extended period of time, not just one-day spikes. And what we've seen in the region, we really sort of noted and started paying closer attention to marine heat waves in 2012, when we had a very large, intense heat wave that spanned from Cape Hatteras all the way over to Iceland into the Labrador Sea. And temperatures in the Gulf of Maine that year were running about 2 to 3 degrees Celsius above the long-term average. This had immediate effects on our lobster fishery in particular, and so really it caught the attention of a number of people and caused us to start really digging into this phenomenon more. So I'm showing you what the 2012 heat wave looked like, but since 2012, we've also had heat waves occur in 2016, 2018, and 2020, and they are becoming increasingly frequent in other regions of the world as well, particularly the North Pacific and Australia. And this figure just shows you for the Northeast Shelf region, the black bars indicate days that we would say we were in marine heat wave status. And for 2020, you see the black lines here, we were in the heat wave status or above that threshold for 207 days of the year. But what you generally notice is that there's a lot more black since 2010, particularly since 2012. So heat waves are becoming more common. They used to be experienced sort of as a surprise in the system, and now they appear to be more of a regular feature of temperatures in the region. So I've given you some insights into temperature change and the nature of temperature change that we're experiencing on the Northeast Shelf. But these changing temperatures can affect fish and invertebrate species in a variety of different ways. I'm going to focus today on sharing with you a bit of a deeper dive into how we're seeing species shift their spatial distributions, how it's affecting population productivity, and also some connections to phenology or timing of events in marine populations. But I also want to point out that the effects can take other forms as well, altering predator-prey interactions, for example, and affecting disease or pathogens that influence a number of these species. So in terms of spatial distribution, generally across the region, we have seen many stocks move poleward or northward in this region and move into deeper depths as they attempt to track preferred temperatures. So this figure shows silver hake back in 1968 from the Northeast Fisheries Science Center's trawl survey. You see them concentrated here off of Long Island at the shelf break. But by the 2000s, showing you here 2014, really concentrated in the Gulf of Maine. So a number of species show very similar patterns. And for the past, decade or number of years, we have been increasingly seeing in our surveys and hearing from interested citizens about many different mid-Atlantic species moving into the Gulf of Maine. Black sea bass is one example, longfin squid, which used to be ephemeral and is now more consistently observed in the Gulf of Maine, as well as a variety of, random species that people report single occurrences of. And this shift in center of populations is also affecting fisheries. So one example relates to the summer flounder fishery. As the summer flounder stock has shifted northward, we've also seen the fishery shift from being concentrated off of Chesapeake Bay and North Carolina back in the late 1990s to being more concentrated off of Long Island Sound by the 2010s. So it goes from the environmental conditions through populations to fisheries as well. When we look ahead to think about how conditions might change in the future and what this might mean for the distribution of a variety of different species, we can put together projections. In this case, I'm sharing with you some projections that draw from the CMIP-5 climate model ensemble using the RCP 8.5 mean, the RCP 8.5 scenario and the mean of the ensemble that reflects that scenario. The 8.5 scenario assumes that we continue carbon emissions as in sort of a business as usual mode. So not substantial policy changes to alter those emissions. And these are projected out to mid-century to 2055. So for a number of different species, about 52 species total, we have looked at how temperature shifts that we expect in the future will alter where these species are located. So we can project the population for a present period and then project it to 2055 and look at the difference in where the species occurs. So in the difference map, what you see in purples reflects a decline of that species, and what you see in pinks or reds reflects an increase. So this gives you a sense of how we expect lobster distributions to change in the future. And as I mentioned, we've done this for a number of species. So you can see for lobster in the fall, we expect to see potential increases mid-coast Maine north into the Bay of Fundy, but declines further south. For longfin squid, we're projecting increases in the Gulf of Maine and on George's Bank. And for black sea bass, increases moving around Cape Cod and a bit off of the coast of Maine as well. When we put the projections together across the variety of species that we've looked at, what we really see is that we are projecting declines in a number of our groundfish species, which are already at the southern extent of their range in many cases. And where we do see increases, we are projecting stronger increases in the Gulf of Maine than in southern New England and the mid-Atlantic, just because the temperature, those have the opportunity to move into cooler temperature areas. They're cooler now to track the temperatures they're currently experiencing. So shifting temperatures also affects the productivity of different populations. And we have seen productivity shifts across a number of commercially important species in the region, declines in the productivity of cod and northern shrimp that can be related to temperature, and then a mixed story around lobster that I'm going to dig into a bit more today. So lobster has been the most valuable single species fishery in the US since 2014, but we see really interesting contrasting trends in populations and fishery landings in southern New England and the Gulf of Maine that our work has indicated are related to temperature. So the Gulf of Maine stock has increased over time, sort of a steady upward trend here until around 2010, 2012, a bit of a decline. Whereas southern New England, in contrast, and many of you have lived through and experienced this, the population experienced a substantial decline in the late 1990s and has continued declining since then. So our goal is to try to understand drivers of these changes. And we did this by sort of asking questions: if you inverted conditions and changed the conditions they experience, what might the population trends have looked like? So in the Gulf of Maine, we see that if the model runs with the actual temperatures, it tracks the population as it changed over time fairly closely. Whereas if we had flipped the temperature and sort of stabilized at a lower temperature level that reflected the average temperature, we would have expected the population to have been lower. In contrast, in southern New England, when you have the stock experience the actual temperatures, the decline that's projected is stronger than if an average temperature over time had been used. And so what this essentially indicates is that the southern New England population of lobster was substantially affected by the warm temperatures that have occurred and still exist in that region. Whereas in the Gulf of Maine, temperatures warmed into a really, ideal zone for lobster productivity, and the population has benefited from that warming. We also considered fishing practices and conservation measures. And interestingly, in the Gulf of Maine, they have protected the large lobsters and female lobsters for a long period of time. If that conservation measure hadn't been in place, we would have expected the population to have been lower than it actually is today. Whereas in southern New England, if the similar protections had been in place to protect the large lobsters, we may have seen the population stabilize at higher levels than under the temperature scenario alone. So I think I just want to highlight that it's important to consider both temperature and fishery management measures and the interplay between those two, as they both do affect the trajectory of fish stocks. And we project this out further, and over time, as temperatures warm, we project declines to occur in the Gulf of Maine stock as we move past that really sweet spot of ideal temperatures. And then in southern New England, some potential for a bit of a rebound depending on the strength and magnitude of warming in the future. So I know that I have to move a bit faster, so I'm going to skip over a couple of things. But one last thing I want to highlight is that changing temperature phenology can also affect the phenology of events occurring across many different species in the ecosystem. So Michelle Staudinger and I led a review of shifts in phenology in the Gulf of Maine, and we saw changes in the timing of phytoplankton blooms, peak copepod, zooplankton abundance, changes in the timing of fish migrations, of seabird egg laying and fledging, and of some of our fisheries. So we see changes that are occurring across many ecosystem levels. And also looking a bit more deeply into shifts in or relationships to fish populations. I neglected to put on the slide a citation for this work, but Liz Henderson, who was a student with Janet and I, worked on a project with us and led this work where she looked at changes in temperature phenology and related it to spatial distribution of marine fish species as well as population relative biomass. And so what we saw is that the spring transition date was related to the spring center biomass of some species, particularly those whose migration might be cued by the spring warming, and summer duration, or how long summer lasted, was also related to the fall center of biomass. And then even more strongly than an indication of change in potential migration timing, we saw relationships to relative biomass of different species. So if summer lasts longer, there may be a longer growing season that could benefit certain species, but for cold water species, it may mean a longer period of stress. So some species experience reduced biomass, or we saw reduced biomass associated with those longer summer-like periods. I don't have time to dig into details on this, but I do want to just spend a couple of minutes reiterating key points that I have tried to get across today. We're seeing a variety of different forms of temperature change through warming trends, shifts in our seasonal periods, particularly the summer, what feels like summer in the region, and heat wave occurrence. We're seeing these affect fish and invertebrates through changes in distribution, population productivity, and the phenology or timing of events that are being experienced by those species. And as I mentioned earlier, I have been very focused on temperature, but I want to just acknowledge that there are many other physical and ecosystem changes that are driven by climate change that can be particularly important in estuaries as well as in the ocean, and that can affect fish populations directly, but also really important to consider indirect effects. So I focused heavily on direct effects in my presentation today, but thinking about how certain changes like sea level rise might affect the abundance and availability and quality of habitat, and how changes in habitat affect fish vital rates and their productivity is really important to bring all of those pieces together. And also to, particularly in an estuarine system, to really think across the system. So trying to understand how precipitation changes in the watershed might affect in river water conditions and how that plays out through the estuarine gradient is also particularly important. So I hope we can through other talks and other conversations, start to put together some of these connections a bit more than I had time to do today, but thank you.
Okay, thank you. Thank you, Kathy. So if you have any questions for Kathy, just make sure you put them in the Q&A session and we'll get to as many of them as we can. So next up, we have Christopher Knack. Mr. Knack received his bachelor's and master's of science from ESF, and he tells me he is now very close to completing his PhD. So congratulations for getting this far, and best of luck to finish it soon. He currently works at Rambo as a scientist, where he has provided technical and writing support on hydrodynamic and sediment flux assessments of ecological restoration. He also works on assessments of land and water use on aquatic systems. He works on the New York State Lake Ontario Resiliency and Economic Development Initiative, and he's been working on the New York State Harmful Algal Bloom Action Initiative. Mr. Knack is an ecologist with a focus on habitat restoration and fisheries management. He has published several manuscripts related to aquatic ecology and habitat restoration over the past 10 years with a wide range of aquatic ecology topics. He's recipient of the 2019 Best Paper Award from the American Fisheries Society Marine and Coastal Fisheries Journal. In addition to his research experience, Mr. Knack has a long family history in the fisheries industry as a legacy commercial fisherman on the Hudson River estuary. So with that, Mr. Knack, take it away. Thank you.
Thanks, Chuck, for the introduction. Before I dive into the science of the presentation, I'd like to talk a little bit about why I'm interested in this topic. As many of you may know, especially environmentalists or fishermen, there's this kind of traditional knowledge that runs of fish are associated with spring blooms, especially the dogwood, which is usually associated with American shad and striped bass. And growing up, this always really fascinated me as being out on the Hudson River with my family in the spring fishing. We always talked about the blooms, not only the dogwood, but there was also something we called lilac shad, which were kind of smaller females that would come in when the lilac started blooming. And this has always fascinated me. And I've always wondered what the connection was between the fish and the trees. And the answer is really there isn't much of a connection between the two. You know, the blooms don't happen because the fish run in. It's really due to more environmental conditions, particularly the phenology between the two species. So the term phenology is really the study of the cyclical or seasonal nature of these plants and animals, and kind of this is really driven by many factors, including temperature, which Kathy talked quite a bit about, but as well as precipitation, day length, moon cycles, many other factors, some probably even unknown. And this is really affected by climate change, especially temperature, which is kind of the most important one. The relationship between temperature and spawning is well established in the literature and is often being the main driver of spawning runs of anadromous fish into coastal rivers, as well as spawning of inland fishes. For example, the yellow perch spawning season occurs approximately 5 days earlier in recent years, as shown by the shifting and earlier season of the midpoint here. Leon's indicated that the shift was primarily associated with warmer spring temperatures. And this has also been seen in anadromous fishes. There's many studies out there that have looked at spawning runs of river herring and American shad, and also in Europe, primarily in France, they've looked at Alosa shad, which is kind of the cousin to American shad. And here's an example from Albemarle Sound in North Carolina. Alewife, they found that alewife spawned approximately 16 days earlier and left the river approximately 27 days earlier. And this was really a shift in about a half a month for the species, for the spawning season. And this has a huge effect on the ecology of the system. And this temperature spawning relationship is really driven by the physiological and bioenergetic processes within the fish, primarily hormones that drive the instinct of these fish to swim upriver, and also for the eggs to ripen. So as the temperature warms, the eggs start ripening faster, and the fish start spawning faster and faster. And what's really important in these studies is the long-term data, primarily water temperature series, and also monitoring of the fish. And we're lucky here in the Hudson River that we have both. There's a historic water temperature series that goes back to 1921. It started at the Poughkeepsie Water Treatment Facility in Poughkeepsie, and in more recent years has been taken over by the United States Geological Survey. There's also a long history of monitoring. There's the Hudson River Monitoring Program, which is also called the Utilities Data Set. And this really came about because of intakes along the river bringing in large quantities of water in trapping and training fish eggs and larvae. So there was a regulatory compliance that utility companies needed to sponsor research on the river. And one of the things that they sponsored was this long-term monitoring program that dates back to 1976. Though it has recently ended in 2017, primarily due to many of the intakes closing. I think the last one closed in 2017. And these large data sets are very important at looking back at the history of the river and how it's changed, especially in relation to water temperature. In 2011, Siegel and Pace published a paper on how the water temperature has changed, and they found that since the 1950s, the water temperature has changed almost 1 degree, and the change in water temperature was greatest in spring and summer. And this is very important because in the springtime, that's when many of the anadromous fish and other fishes, important fishes in the river, start to spawn, particularly American shad and striped bass. And another interesting thing that they saw, if you look in the graph on the right there, is that in the early 1900s, the river was warmer, and that's usually assumed because there was deforestation in the upper parts of the river, particularly in the Adirondacks. So in 2019, we published a paper looking at the changes in American shad and striped bass spawning seasons in the Hudson River, and we looked at the historic spawning as well as projected it out to 2099 using data from the International Panel of Climate Change, similar to what Kathy showed, the RCP 8.5, which was the standard, and also some of the other scenarios that they have. The key findings of this study showed that American shad and striped bass were predicted to spawn about a month earlier in 2099 than they did in 1950. And in 2099, they're likely to have a similar spawning season to what we now see in the Chesapeake Bay. And historically, they spawned about 5 days later into the spring. And this is really driven by the spring warm up or transition. So now the springs are warming up quite a bit faster than they were 40, 50 years ago. And that's really driving this shift of about a week in the spawning season. We also predicted that the spawning season would likely be reduced up to about 20% by 2099. And this doesn't sound a lot because it's about 10 days of their spawning season, but this could have large ecological effects and effects on year class, in that if a large storm event came through or some other event happened that affected a number of the larvae and eggs, they may not have time to recover as if they did if they had a longer spawning season. And there's many different morphological, ecological, and changes that may occur as these fish start spawning earlier in the season. One is kind of a reduced spawning size. It's well known that particularly American shad, as you move south along the Atlantic coast, the shad tend to get smaller and smaller. Though they typically have more eggs per fish, which is kind of you think they would have smaller or fewer eggs, but they typically have more eggs, which kind of makes up for their smaller size. And they also typically respawn fewer times. And that means coming back after they've already spawned once. So in the Hudson River, there's records that American shad have come back at least 8 times and spawned, where down in the south, they're actually semiparous like a salmon, and they only spawn. They just come in, spawn once, and die. It's not likely that the Hudson River shad population would ever be semiparous, but they couldn't. We could see a reduction in the number of repeat spawners. There's also likely to be some ecological changes, such as the predator-prey mismatch, which Kathy talked a little bit about, or at least touched on. And this is really where the timing of the spawning is related to the bloom of zooplankton in the spring. And it's typically timed that way because when larval fish first absorb their yolk sac, they need to feed relatively quickly or they're likely to die. So if they come in and they spawn and the zooplankton aren't there, you're likely to get a little bit more mortality than you would expect. And as I mentioned, there's also a greater impact of weather-related mortality. If a storm comes in during peak spawning and causes increased mortality of eggs and larvae, you're likely to get a reduced year class. And this could affect the population going forward. It's also possible that you might see some extirpations or introductions of fish in the future. As Kathy talked about, many species are shifting northward. We've already seen the loss of one species in the Hudson River, the rainbow smelt, which used to be a fairly common sight a few decades ago. Now their range is considered to be north of Long Island. So this is kind of the map of the DEC here, showing that rainbow smelt are in the river, but they have not really been seen for a couple decades. You might also see the loss of some brackish and freshwater species, which also don't get that luxury of being able to move north as things warm up. And as Scott George is going to talk about in a little bit, there's also likely going to be some invasions as the water temperature warms up. Some species from the south, either native to North America or introduced, will be able to spawn in the Hudson River, and you'll likely see their populations increase or show up. And in the future, it's really important to continue research in the Hudson River on fish, specifically spawning phenology. And this includes the species-specific community and whole ecosystem studies. And to do this, we really need data, a lot of data. As Kathy showed, many of the research project that she highlighted showed a lot of data and over a long period of time. And that is really important. So we need to continue to monitor the fish species, which the DEC has several monitoring programs and are hopefully going to expand some of those in the future. And short-term studies are also very important. So once you get these long-term trends, sometimes these short-term studies are very important to determine how or what you're seeing. And funding opportunities are also of great importance to bring in new researchers and to highlight some of these shorter studies. So a couple that are kind of coming up right now are the Mark Bain Graduate Fellowship and the T. Polgar Fellowship. And this kind of helps bring new people into the fisheries community in the Hudson River and expand our total understanding of what's happening out in the river. The DEC does a great job monitoring, but sometimes they have a hard time doing some of these short-term studies. And with that, I think I talked fast, so I caught up some of Kathy's time, and thank you.
Okay, thank you very much, Chris. So I'm now going to find my notes. And next, I'm going to introduce Stuart Finley. So Dr. Finley received his PhD from the University of Georgia way back when. And he's currently emeritus at the Carey Institute of Ecosystem Studies, and he chairs the Hudson River Estuary Management Advisory Committee, a committee which he has served for many years. His research interests span habitats and ecological processes in aquatic ecosystems with a strong emphasis on bringing information to management and applications. He has studied streams, wetlands, and rivers broadly with a focus on the Hudson Valley of New York. He received the Outstanding Environmental Researcher of the Year Award from the Hudson River Environmental Society. He has been studying Hudson River submerged aquatic vegetation and freshwater tidal habitats for more than 30 years now, at least as long as I've known him, which has been about 30 years now, Stuart. He brings a wealth of experience and knowledge to us today. So thank you very much, Stuart. Take it away.
Okay, thanks, Chuck, and thanks everybody else for being here, and to the organizers of this symposium and HRF for helping to pull it all together. So I'm going to talk more about where some of these organisms live rather than the vertebrate organisms themselves, the fishes. And I'm also going to talk about sort of what these places do, what they do ecologically, what some of the functions and other values are that these habitats or places provide to the things that live in the river as well as us. Okay, now my screen is frozen. This is great. There we go. So I'm going to talk about climate effects as I was asked, and I'm going to break these into a couple of different categories. There are many aspects of climate that happen slowly and steadily. Temperature you've heard about quite a bit in the last two talks. The other two that fall into this sort of slow and steady category are sea level rise and salinity intrusion. And then some climate effects are a little more dramatic and represent step changes. And I'll talk about storms, which are pretty clearly dramatic. And then earlier in the first session, John Waldman talked about some of the really big sort of global climate drivers, North Atlantic oscillation. I don't think we get really big El Nino effects here in the Hudson, but he showed that the NAO does matter. So in order to talk about sea level rise in some of these shallow water nearshore habitats, it's important to recognize that the vegetation we see out there is driven by the inundation regime. So there's a whole group of plants, mostly sort of grass-like, and on this slide I call them graminoid, quote unquote, that occur at the highest elevations that receive some level of tidal inundations. And then a little bit lower, there's a zone of what are called the broadleaf plants. And this is largely Nuphar advena, although there's a variety of other things in there. And these occur about 60 centimeters lower than the higher tidal plants. And one of the things I'll be doing is contrasting what we know about the extent, sensitivity to inundation and functions of these two classes of vegetation. We'll come back near the end and talk about the submerged plants, which are completely underwater almost all the time. So the first topic is these two classes of plants that occur at slightly different elevations. And so what's going to happen as sea level rises? It's coincidental but important that the difference in elevation between these two zones of plants is about the same as what we anticipate for sea level rise by near the end of this century. And so the figure here is showing the percent cover for one particular site. It happens to be Tivoli North Bay. the percent cover by these high intertidal grassy plants over about a 70-year time span as sea level rises. And what you see, and is not surprising, is that as the inundation regime gets deeper and deeper, so more frequent flooding that lasts longer, these plants that aren't used to that, so to speak, are replaced by those that have lived for quite a long period of time with a more frequent and deeper inundation regime. So essentially, the high intertidal plants will be replaced by these low intertidal plants. And the modeling that was done for Tivoli Bay, shown in the graph here, has been essentially verified by a different kind of model that spanned the entire river. And this was done by Navatabak and colleagues at Scenic Hudson. And Chris Solomon talked about that a little bit in the first session of this seminar series. So we've got a change in composition that we expect to see here. So what difference does it make to what these habitats are doing ecologically? It turns out that much of what they do ecologically is dictated by what kind of plants are growing there. And I just showed you that the kind of plants that are growing there are dictated by the inundation regime. So the things, the ecological processes or functions that are associated with these high intertidal grassy plants, we stand a real chance of losing many of those. And we know quite clearly for 3 of the 4 functions listed here that the high marsh actually provides these values to a greater extent than the broadleaf that occurs lower in the tidal window. So there are good data showing that there's more nitrogen removal, a sort of pollution abatement function that occurs when you have more of this high marsh present, and less of that occurs when the marsh happens to be dominated by these broadleaf plants. Clearly different habitat values. There's quite a lot of data that Chris has contributed to, as well as a variety of other folks, Eric Kiviat, Bob Schmidt, and others, showing exactly which fishes use the different habitat types. And certainly for organisms, be they amphibians, reptiles, or other things, they're mammals or birds that don't like to be wet all the time. And so for those particular organisms, the high marsh that's not inundated quite as frequently or for as long provides greater habitat value for those things. The high marsh with these grassy type plants provides greater storm protection. So there's a lot of interest in how the coastal wetlands or the Hudson River wetlands help surrounding human communities. And for those of you that know the story of Piermont Marsh, they value their high marsh a great deal. And it's been shown recently that it really helps dampen down debris transmission during storm surges and things like that. There's a lot of interest right now in carbon sequestration and how important wetlands in general, or different kinds of wetlands, are in tying up carbon. If I had to guess right now, I'd say the high marsh is doing more of that than the low marsh is. Although our data on the low marsh is not really all that great, and there are reasons that it might be doing better than I'm guessing. But if you asked me right now and I had to guess, I'd say the high marsh is doing better than the low marsh. So here's a series of 4 things that would change dramatically if in fact we went through a transition from high marsh to low marsh, as sea level rise sort of changes the suitability of those places for these various plants. So what's going to happen in the bigger picture as sea level rises? There are several options for what might occur. The bottom pieces of this panel show one possibility, and that is that as sea level rises, these marshes will accumulate sediment and grow vertically and essentially keep pace. They will get taller, so to speak, as the water levels rise. And we kind of hope that's the case. This is what has happened since the glaciers left and sea level has been rising at what I'll call sort of a normal rate. Some wetlands have a chance to move horizontally and can migrate inland. And where that is possible, various entities, DEC and Scenic Hudson and others, have tried to make sure that their path for movement is clear. And this can happen. This is not something that I anticipate will happen commonly with Hudson wetlands. They're constrained by either human barriers, as shown in the bottom, or just the naturally steep topography of the Hudson Basin. So one of the really critical things is, what is the opportunity for vertical growth in these wetlands? And I'm going to show you a couple of data slides in a second, but just to give you some perspective, these things to keep pace with sea level rise are going to have to grow as fast as they are now, as fast as sea level is rising now, which is about 3 millimeters, 0.3 centimeters per year, to as much as 2 centimeters a year, which is a reasonable projection for the end of the century. So how are they doing now? And how do we know that? And there are a bunch of ways to estimating how quickly wetlands are growing vertically. There's a very nice technique that's been pioneered by folks at Lamont, where you look at lead going down into the sediment. You look at how much lead is in different levels, layers going down into the sediment. And fortunately or not, humans started putting lead liberally into the environment at roughly 1900. So if you see elevated lead, it means that those sediments were deposited after 1900. And this figure shows you that this particular profile of lead, distribution of lead, came out of the gray box there on the left as sort of the ambient pre-human lead levels. And so this core, this profile started showing elevated lead at about 60 centimeters. So what it's saying is that those 60 centimeters of sediment above that have been deposited in the last 120 years. And conveniently, the arithmetic works out to half a centimeter a year. So this is how you can determine how fast these things are growing vertically by looking at lead. So we've done this for a lot of sites up and down the Hudson, and what we see, not surprisingly, is a pretty big range in how fast these things are growing vertically. There are a number of sites that are growing slowly, and that's indicated by the figure on the left, which is Iona Island, for those of you that know it, just south of the Bear Mountain Bridge. And lead doesn't come sort of above pre-human, pre-industrial, I should say, not pre-human, pre-industrial levels at about 30 centimeters. So those 30 centimeters have been deposited since 1900. It gives you about 1/4 centimeter per year. And that's about what sea level rise has been over the last few hundred years. And then we have some sites that are showing amazingly high deposition. And so the site on the left is Hudson North Bay, just north of the city of Hudson. And we don't get back to pre-industrial lead at the bottom of the core, and that's a meter-long core. So at least 100 centimeters have been deposited in 120 years. And that's a pretty good 3 tons faster than sea level rise. And so we've got some sites that look like they're holding on right now, such as Iona, and we've got some sites that are growing plenty fast, such as Hudson North Bay. Fortunately, there's some pattern here, which leads us to ask, what's driving these patterns. But the slow sites tend to be in the southern parts, exemplified by Iona Marsh. And the really fast sites seem to be up north. So this pattern makes sense because the big sediment delivery that's supporting this vertical growth is coming in from the Mohawk and the upper Hudson. So that makes sense. You always have to be a little skeptical as to whether what makes sense is really true, but that's okay. It seems to make sense, but it does give us some way of allocating option for doing some management, because some of these sites pretty clearly could use some help in accreting vertically, growing vertically. Others maybe don't need so much help, so we can sort of put our effort where it's best needed. Is the pattern really just geography, or is there something else going on? If there's something else going on, we haven't found it yet, but you're always wise to consider that there could be something else going on. And is there enough sediment? At some point, if they have to grow at 2 centimeters a year for decades to hundreds of years in the future, are they going to run out of sediment delivery at some point? And right now, we don't know that, but we could probably try and do some estimates. So the other slow change that's related to climate I want to talk about is salinity intrusion. This happens every day. It happens over the seasons in very well understood and predictable ways. Most of the variability is in the highlands. And so there's been a site operating, got it, see you, Jim. There's a site that's been operating at West Point measuring salinity for quite a long time. And as you heard earlier from the Karin Lindberg and John Waldman, salinity has large effects on the fishes and a variety of other organisms. So we've got this sort of balance between sea level rise, pushing that brackish water where the seawater is, pushing that northward inland, and increased freshwater flow, pushing it out towards the harbor and out towards the coast. So you got this tension between these two forces. Sea level rise has risen, freshwater flow has increased. So where is the sort of balance between these two forces as of right now? A couple different data sets to show you. The one at the top is from Kerry. We visit sites in the boat and grab samples 4 times a year. So that top time trend is from August, when you expect the greatest intrusion of sea level, because that's a relatively dry time of year. I see no trend there at all. That winds up being 1 sample a year for 30 years, so it's a long time span, but not very frequent. The panel at the bottom is from the USGS salt front study, as well as more recently from RICOS, and that's every 15 minutes for 15 or 16 years, so a lot more data, but neither one really shows a trend. So as of right now, it looks like we're sort of at a stalemate on this tension between sea level rise and freshwater flow, and we'll see how that plays out in the next, in some period of time. We don't know when. The last thing I want to talk about are the submersed plants that are underwater. And the predominant one is this grassy looking thing. It's Vallisneria or water celery. And I want to talk about a step change or a pulse disturbance, and that's Hurricane Irene, which I imagine many people remember, happened very late in the growing season 2011. And if you look at the picture at the lower right, all the flow and sediment coming down the Mohawk, it's probably not surprising that the submerged vegetation in the river really took it on the chin. They don't have a chin, but if they did, they'd take it on the chin. So the data are from a team of volunteers that go out every year and monitor sites. And what they showed us is what we heard from a lot of anecdotal information, as well as aerial photography surveys. Right after the growing season following Irene, the abundance of extent of SAV was way down. And it's probably fair to say the last year or two, we've kind of recovered to where we were. But that's a long time. That's 6 or 8 years without a really key habitat that's known to be important for a number of small fishes. It is natural recovery, or are we okay with waiting 6 to 8 years for this to recover? Or should we be prepared to do something about it? So to wrap up, there's a lot of news here. Partly, we do know a lot about the Hudson, and Chris just talked about some of the good monitoring studies that we've had, some of which we're standing a likelihood of losing, I'll point out. But we do know a lot. Many of these climate consequences are predictable. That doesn't mean we like them, but it means they are at least predictable, so we know what we might lose or what might be at risk. Some of these things can be mitigated by management and planning, some cannot. But if we're ahead of the game a bit, we might be able to get in front of some of these changes. Part of what we're supposed to do in this series is talk about what are the open questions that could stand some attention. And we'll get some change in the extent of these two kinds of wetlands, the lower intertidal, upper intertidal kinds of things. And exactly, what are we going to gain, what are we going to lose, and when is pretty key, and we don't have a really good handle on that. At some point, salinity is going to break through. We don't know when. We don't know if that's going to be slow and steady, or is that going to be, 1 or 2 dry years back to back is really going to lead to a step change in what's going on. We don't know about that. Good opportunity for some modeling here, I suspect. And then lastly, this SAV recovery. We know we got by with natural recovery the first time. Are we prepared to say, yeah, we'll just let it play out the next time this happens, whenever that may be. I'm in no rush to see that again, but whenever that may be. So I'm going to stop just barely under 20 minutes so Jim doesn't get too excited, and I will stop sharing and hand it back to the next speaker. Thanks, everyone.
Okay, thank you very much, Stuart. So next up we have David Strayer. Dr. Strayer received his bachelor's from Michigan State University and his PhD from Cornell. He too is emeritus at the Cary Institute of Ecosystem Studies, and he's also a visiting scholar at the Graham Sustainability Institute at the University of Michigan. Dr. Strayer's research focus has been on the ecology and impacts of non-native species on freshwater conservation ecology, especially of the freshwater mussels, and on the ecology of the Hudson River. He's a former president of the Society for Freshwater Science, and he too had received the Outstanding Environmental Researcher of the Year award from the Hudson River Environmental Society. So thank you very much, Dave, for speaking today, and you can take it away.
Thank you, Chuck. Let's get this thing going. So I want to talk today about invaders other than fish and how they've affected fish populations in the Hudson River in the past and how they might affect the fish populations in the river in the future. I want to start right off by looking at a few case studies of some recent invaders. The first invader I want to consider is zebra mussels. Zebra mussels are a ballast water invader. They appeared in the Hudson in the early 1990s, and they have been very abundant in the river ever since then. There's several different ways by which zebra mussels might affect fish populations in the river. The first is some fish eat zebra mussels, and I'm thinking particularly of the sturgeons and the freshwater drum. I don't know of any studies looking at how the abundance or growth rates of these fish changed when zebra mussels appeared in the river, but the timing of the arrival and spread of freshwater drum populations in the Hudson River suggests to me that there might be a cause and effect relationship here. Another way that zebra mussels might affect fish in the river is by changing the physical character of environments in the river. So, for example, about 10 square kilometers of the river bottom used to be bare rock up in the freshwater part of the river. These are now covered by zebra mussels and could presumably affect fish spawning and things like that. Again, I don't know of any studies of this, how this might have affected fish in the river. Zebra mussels are filter feeders, and so their activity has also made the Hudson a little bit clearer than it used to be. And we think this has led the submerged aquatic vegetation beds to spread and become more productive. And this ought to favor some of the littoral zone fishes like killifishes and sunfishes and so forth. And I'll be showing you some data on that in just a minute. The most important way, though, that zebra mussels probably are affecting fish in the Hudson River estuary is by affecting the food base. Zebra mussels have eaten a lot of plankton since they arrived in the Hudson River, and this has led to a large decline in fish forage in the river. The graph here on the bottom shows the time course of zooplankton biomass in blue and in benthic dense animal densities in red outside the littoral zone of the river since before zebra mussels arrived and then early invasion and late invasion. And you can see that when zebra mussels appeared in the early 1990s, the amount of fish food in the river fell by about 50%. So there's a lot less for the fish to eat than there used to be, especially out in the open water of the river. You'll also notice there's a partial recovery of those forage invertebrates later in the invasion. I don't have time today to talk about that, I'm sorry. I'm going to be focusing on that first 10 years or so of the early invasion period. Based on what I just told you, you might hypothesize that the fish that live in the open water of the river and feed on open water invertebrates, things like the aloses and the moronis, might suffer as a result of the zebra mussel invasion, and whereas the littoral zone fishes might flourish after zebra mussels arrived. And by suffer and flourish, I mean that the abundance of these fish might change, their growth rates might change, and their distribution within the river might change as well. I was able to work with some actual fish biologists to analyze the long-term data sets that DEC and the utilities have collected to see whether we could in fact detect and affect the zebra mussels on fish distribution, abundance, and growth. So this slide's a little busy. On the x-axis, we have the different kind of fish species. We're looking only at young of year for these fish species. On the y-axis is the change that we estimate occurred after zebra mussels arrived in the river. The dots are the mean estimate, and the whiskers show various confidence limits. The broad whiskers are 90% confidence limits. The highlighted yellow areas are the zones where we expect the data to lie if our hypothesis is correct. That is, littoral species should become more abundant, shift upriver into the area where zebra mussels occur, and grow faster, and the open water fish species should do exactly the opposite. I think you can see a pretty clear pattern with fish distribution and growth. It pretty much follows our hypothesis. Most of the data points fall in the yellow zone and very few in the white zone. With abundance, the picture is less clear. Most of the littoral zone fish species seem to have increased in abundance after zebra mussels arrived. The picture is more mixed with the open water fish species, which are of course of the greatest interest, I think, to people. And you might conclude from these graphs that zebra mussels affected fish distribution and growth, but did not affect fish abundance. I think that's really the wrong interpretation. The correct interpretation is we can't tell whether zebra mussels affected fish abundance. Notice that these confidence limits span a range of 3 to 10-fold in fish abundance. So we really can't tell whether the fish abundance changed, especially for the open water fish species. The second invader I want to talk about is the water chestnut. This is a plant that was brought in deliberately as an ornamental plant into North America from Europe, and appeared in the Hudson in the 20s and 30s, and it now covers about 600 hectares of the river shallows. These two unfortunate people are trying to make their way through a water chestnut bed here. The dominant effects, likely effects of water chestnut on fish have to do with its physical engineering of habitats in the shallows. These water chestnut beds are so dense that it's almost like someone put a great big tarp over the river shallows. Only about 1% of sunlight is able to penetrate through these water chestnut canopies, and they pretty much block off atmospheric exchange of gases between the water and the overlying atmosphere. And as a result, the oxygen dynamics in these beds are dramatically different than they are anywhere else in the river. The graph near the top of the page here is from Nina Caraco and her colleagues from the Carey Institute. The blue line in the middle shows the oxygen concentrations in the open water of the river over the summer and early fall. The green data are from a Vallisneria bed, a wild celery bed, and you see the oxygen's there's a little more oxygen in these beds as a result of plant photosynthesis than there is in the open water of the river. The red dots are data from a water chestnut bed, and you should see two things right away. The oxygen is all over the place. It goes from maybe 8 parts per million down to 0. And for much of the time, the oxygen in these water chestnut beds is below 3 or 4 parts per million, which we think of as being stressful for fish. And in fact, in a lot of times, the oxygen literally reaches 0. So this should be bad for fish. The other way that water chestnuts might affect fish is, again, through their effect on the forage base. And on the graph at the bottom of the page here shows the number of macroinvertebrates in unvegetated areas, in water chestnut beds, and in wild celery beds. And you can see that there's a lot more in macroinvertebrates in the water chestnut beds than in the other habitats in the river. So these are sort of a very rich cafeteria for fish. And by the way, the invertebrates should have just as much trouble with low oxygen as the fish should. So this is interesting that the invertebrates are able to do fine in these periodically deoxygenated habitats. So how would you figure out what effects water chestnut have on fish? Well, one way, if we'd been smart, we could have studied fish during periods of changing water chestnut abundance. So during the period of initial spread in the Hudson, or during the 60s when the state was spraying a lot of 2,4-D on these beds to try to eliminate them, we could have studied fish populations at the same time to see how they responded to changing water chestnut areas. No one, I don't think anyone thought to do this, and I'm not aware of any data sets that we could use to do this sort of analysis. Another way that you could look at the effects of water chestnuts on fish would be to go into a water chestnut bed and see what kind of fish live there. And I hope that these pictures of Bob Schmidt and one of his students convince you that this is really not a very easy thing to do. This is a lot of work, and it's not very nice work. But it's something that Bob and his students have done in several water chestnut beds in the Hudson. And again, based on what I showed you of oxygen dynamics in these beds, you'd expect there either to be no fish in these water chestnut beds, or you might expect there to be a peculiar community of fish species that are adapted in some way to deal with these low oxygen concentrations. What you see instead, though, the pie charts show the species composition of fish in different water chestnut beds, 5 different water chestnut beds up and down the Hudson. And without getting into the details, what you see is there are, in fact, many fish in the water chestnut beds, and that the different beds contain different species of fish, including many or most of the fish species that you would expect to find in the river shallows. So we're left at this point, even though there are dramatic effects of water chestnut on the environment in the shallow water of the river, we don't have a clear idea what they're doing to fish communities. The third case study I want to mention briefly is of this microsporidian fungal parasite called Glugia hertwigi. This is a parasite, a fish that's attacks smelt primarily. It's native to Europe. It appeared in North America in a number of different sites in the mid-20th century. And it's associated with dramatic die-offs of smelt in the Great Lakes, in some inland lakes, and in some of the northeastern estuaries as well. The utilities biologists a few years ago were interested in whether this parasite might have been partly responsible for the disappearance of smelt that Chris Knack just mentioned. And so you can see the graph on the bottom shows they went back to archive samples and they looked for this parasite in samples of smelt from the Hudson in 1991 to 1995, just before smelt disappeared from the river. And you can see that by the end of their first year, almost all of the young of year smelt in the Hudson were infested with this parasite. So you've already seen this graph on the left, the dramatic disappearance of smelt from the Hudson in the mid-1990s. This is conventionally explained by rising water temperatures. We have a wonderful temperature record of water temperatures in the Hudson. But you can also imagine that this disappearance might have been explained by the high prevalence of this parasite, which is known to cause smelt die-offs at other sites. So at this point, we don't know to what extent temperature, this parasite, or their interactions are responsible for the disappearance of smelt from the Hudson. And I would caution everyone not to be seduced by the wonderful temperature record, and assuming that the explanatory variable that you have more data on is the one that explains what you're seeing in nature. All right, so I want to look briefly. So here's my conclusions from the case studies. I hope I've made the point that non-fish invaders affect fish through multiple pathways. Rarely do we have good estimates of such effects on fish. And nevertheless, I think that the available evidence that we do have suggests that these non-fish invasions may have had large impacts on the Hudson River fish communities. There are other invaders besides the ones that I mentioned. The graph on the left here is updated from a study that Mills et al. Did in the mid-1990s, showing the number of established invaders in fresh waters of the Hudson River Basin. You can see we now have about 150 known established invaders, about a quarter of them fish, 3/4 of them not fish. These data surely are underestimates because they overlook many small organisms, and they don't consider species south of the Tappan Zee Bridge in the sort of more marine district. The current invasion rate is about one new species per year, and we have a number of past invaders besides the ones that I mentioned that are capable of strong impacts on fish. All right, so let's turn our attention to the future in my remaining few minutes here. What can we say about the future? Well, it's a very easy prediction to say that we can expect many more invaders to establish in the Hudson. And there's two reasons that I would make this prediction. The first is simply an extrapolation of the graph on the left. We're now seeing invasions at about one per year, and in 30 years, we can expect 30 more invaders. That seems pretty straightforward. The other reason I can make this prediction is that we know the invasion pathways that are bringing new species into the Hudson River watershed. And by and large, these are under incomplete or poor control. So until we control these invasion pathways, we can expect to see more invaders. We can also predict that some of these invaders will have large effects on fish. I listed a few examples of species that I expect to appear in the Hudson in the coming few decades that might affect fish populations. But I would also emphasize there are a bunch of invaders that will come into the Hudson that we don't know, that we would not currently understand them well enough to predict that they would have large effects on fish. And this is particularly true of fish pathogens, where we're still seeing a lot of emerging diseases. Now, I would say that this future prediction, the prediction of the future I just mentioned, does not have to be. We can control invasions if we choose to do so. And there's a very nice example that I'm showing you here from the Great Lakes. On the x-axis here is time, on the y-axis is the cumulative number of shipping-related invaders that have come into the Great Lakes and established themselves since the opening of the St. Lawrence Seaway in 1959. And what you can see is an exponential rise in the number of shipping-related invaders coming into the late 80s and early 1990s. When the zebra mussel appeared in the Great Lakes in the 80s, particularly, there was a lot of concern in the Great Lakes community that this had to be stopped. And so the provinces, Canadian provinces and the US states got together and adopted strong ballast water controls. And you can see that they've pretty much shut down the arrival and establishment of new shipping-related invaders into the Great Lakes. What else can we know about the future? We know that invasions are going to interact with other drivers of the future Hudson, and the previous speakers in this session have set this up very nicely. We're seeing climate change. We're going to see a dramatic change in temperatures and things like that in the Hudson in the coming decades. This is going to allow invaders from warmer climates to come in and thrive in the Hudson. And mostly we think about nearby regions like Chesapeake Bay, but I want to encourage you to think about Florida and Asia and distant areas as well as potential sources for new invaders in the Hudson. These invaders will get here both through human means and through natural dispersal. Kathy was talking about natural dispersal in the marine species. For freshwater species, I expect human-aided dispersal is going to be much greater than natural dispersal. Habitat change, which Stuart just talked about, is going to create new opportunities for invaders. And then finally, deliberate management may affect invaders. So Dan Miller was talking about creating new habitats in the Hudson. This will offer opportunities for invaders to inhabit these new habitats. And also we may deliberately control some of the invaders. So for example, for as long as I've been working in the Hudson, people have been talking about trying to get a biological control of water chestnut going. All right, so I'm going to close with this. I think very nice question that Scott George raised on a phone call a couple of weeks ago when we were planning this session. Are we poised to understand and manage the effects of future invaders? And I would rate our ability as sort of mixed. We already know we have plenty of scientific information to reduce future invasion rates. This is a question more of whether we have the political will to apply that scientific information to reduce future invasion rates like they did in the Great Lakes. We generally don't have very good information on how to control established invaders, like things like zebra mussels or glugia, that microsporidium. We don't have good information necessarily on how to control them. We also often do not have good information on predicting the effects of control. So if we decide to go ahead with biological control of water chestnut in the Hudson, what effects will that have on fish communities? I hope I've convinced you that it's likely that there will be large effects on fish communities if we were to eliminate water chestnut from the Hudson, but we just don't know what those effects would be. And then we have a sort of moderate ability to detect new invasions, particularly better for the larger bodied organisms, and to assess the effects of new invasions on the Hudson River fishes. And the ability to detect and assess the effects of new invasions depends greatly on the future of the biological monitoring programs in the Hudson, which already a couple of the speakers have mentioned are in danger of being discontinued or already have been discontinued. So thank you for your attention today.
Thank you very much, Dave. So we're going to move on to invasives part 2, and Scott George is going to speak about invasive fishes. So Mr. George received his Master's in Arts and Social Studies, but then moved on and got a Master's in Science in Ecology and Evolutionary Biology. He's a research biologist with the U.S. Geological Survey at New York, at the Water Science Center in Troy, New York. His research covers a number of topics related to ecosystem disturbance to aquatic ecosystems, including acid deposition, climate change, and invasive species. Most recently, Mr. George has been leading research in the Mohawk River and the New York State Canal System, looking at the eastward expansion of the invasive round goby towards the Hudson River, which I believe we're going to hear more about. So thank you very much, Scott.
Great. Well, thanks, Chuck. And Chuck, can you give me a thumbs up that we're good with the audio and the visual?
I see and hear you.
Perfect. Thank you. Great. Well, thanks. So as Chuck mentioned, my name is Scott George. I'm a research biologist with the USGS in Troy, New York, and I'm flattered by the invitation to present before you today. My talk will be a little bit different because much of it will focus on a case study, the research that Chuck mentioned as we look at the eastward expansion of round goby through the canal system and ultimately towards the Hudson River. And then if I manage my time well, we'll get to talk about a few other fish invaders on the horizon that may also be knocking on the door. So round goby, well known to most folks at this point. This is an invasive benthic fish from the Ponto-Caspian region of Eurasia, first detected in North America in 1990. Round goby quickly colonized all 5 Great Lakes, and genetic analysis indicated that there were likely multiple introduction points, probably via ballast water, that contribute to this rapid spread. When goby colonize new areas, there's a whole host of negative ecological effects that are observed. Goby are very effective at outcompeting our native benthic fish. So our darters and our sculpin in particular really tend to take it on the chin. Additionally, there's a lot of concern about the role goby may have in reducing recruitment of nest building fishes. Goby also, there's concern, can bioaccumulate contaminants and pass them on to higher order consumers due to their heavy diet of dracinid mussels. And finally, goby are a carrier of viral hemorrhagic septicemia and are also believed to be an important vector in some of these avian botulism bird kills that we see on the shores of the Great Lakes. Closer to our study area, round goby were first detected in Oneida Lake in 2013. Cornell researchers determined that only 2 years later, they had become the most abundant benthic fish in the lake. We became interested in 2014 when an angler captured a specimen in the canal system near Utica. And that opened a whole lot of ugly questions about what was the current distribution of round goby at that time, how quickly were they expanding, and when might we expect to see them in new watersheds. So our research has been concerned really with this region represented by the red arrow here. So we're going to call this the Mohawk River portion of the New York State Canal System. And it's a really important section of river because it serves as the gateway through which round goby can potentially reach the Hudson River. Once in the Hudson, of course, they can go north through the upper Hudson and Champlain Canal into Lake Champlain. And more interesting to this group, they can potentially go south downstream into the Hudson River estuary. And the estuary is really interesting, as everyone here knows. Obviously, the lower portion of it is brackish, and the location of the salt front and the concentration of the salinity, of course, varies depending on the ambient flow conditions. But I'm showing you some arbitrary salinity data here from the Piermont monitoring station just to prove a point. So this suggests that salinity is generally around or below 10 ppt. Round Goby have a salinity tolerance of somewhere between 20 and 30 ppt, or PSU, pick your unit of choice. And that depends largely on the dominant salt molecule within that salinity. So what this is telling us is that salinity alone is not going to be enough to prevent Round Goby colonization of the estuary. Other factors, may play a role in reducing the success of Round Goby, but salinity alone is not going to save us, so to speak. So the objectives of our project, we wanted to try to find the best early detection technique for monitoring this rate of expansion, which has been pretty darn fast in some of the Great Lakes states. And we got at this using a comparative methods study, so 3 traditional sampling methods, as well as environmental DNA of water samples. And that's really kind of the, I guess, the trendy, sexy part of this project that people get excited about. I think it's become pretty mainstream at this point, but what are we talking about? Environmental DNA, a nice tidy definition here. You can think of it as genetic material that's obtained from environmental media. So water, sediment, air, who knows? And, as an example, you take a liter of water out of a lake or a river. You don't actually have any fish in that liter of water, but based on the fragments of DNA in that water sample, you can actually infer what fish species or any other organisms were present in that immediate vicinity. And of course, why is there DNA in the water? A whole host of obvious reasons that, surprise no one. So we took all 4 of these gear types and employed them twice annually at 12 sites on the canal system for a 4-year period. You can see the sites range here from Sylvan Beach, right next to Oneida Lake, all the way down to the confluence with the Hudson. We even have one site here in the estuary. And as far as the fish sampling, I'll go quickly through this. Seining, 30-foot bag seine, 3 poles at each site, rotating around a fixed point on shore. Trapping, using G minnow traps deployed in pairs for a 1-night soak. Trawling, we're using a benthic trawl, a Siamese trawl, which is comparable to a mini Missouri trawl you may have experience with. And if you haven't pulled a trawl along the bottom of a large river or urban canal, it's pretty exciting. There's a lot of cool anthropogenic and natural items down there just waiting to get captured, many of which are heavily colonized with dracynid mussels. We even had the misfortune of tangling with a few ill-tempered blue crabs in the estuary this past summer. Unfortunately, the trawl is very effective, so we're kind of stuck using it for the long haul, it would seem. So getting into 2020, and I'll show you some results, but a few things I need to tell you. First, trapping and seining, very ineffective methods for sampling Round Goby, at least in the canal environment. So we discontinued them going into 2020. Second, and Karen mentioned this in her talk, the wintertime drawdown in the Mohawk River was maintained well into the summer, I believe into early July. So you can see the struggles that we went through trying to get a boat in the water. The yellow arrow actually represents where the water level normally would be at this boat ramp. But 2020, in many respects was a struggle, and sampling was no different. So 2020 is also important because it kind of marks the point in this project where we sort of lost a confident handle on what was going on with this invasion. Going into 2020, we felt like we had a pretty good handle on where the invasion front was, what the rate of expansion was. We felt like Round Goby were somewhere a little bit east of Utica, probably around Frankfurt or maybe a little bit east of there. And 2020 really turned our research kind of on its head. This being probably the most notable aspect of it. This excited young lady in November of 2020 captured what appears to be a large adult male Round Goby This is between locks 6 and 7, just a few miles from the Waterford Flight, and obviously, therefore, just 3 or 4 miles away from the Hudson River. And this posed a lot of really ugly questions that we debated with our Region 4 DEC counterparts, trying to understand, what does this mean? Is this a bait bucket transfer? Is there an established population at this location? What do we really know about the distribution of goby at this point? So we resampled the heck out of this area in November, did not produce another goby. And then at that point, we kind of waited nervously for the 2020 environmental DNA results to come back to hopefully shed some light on really what this means in terms of the broader watershed. So here's a super dense data slide that we'll walk through together. I have the trawling data on the top, eDNA data on the bottom. All the data is arranged chronologically as shown in the legend. So oldest data is on the top, newest data is on the bottom. And if we start in the top left with the trawling data, these are mean catch rates for each survey. And you can see out close to Oneida Lake in the canal, we're catching goby in almost every survey, often at high densities. Go over to the Rome location. For 5 surveys, we don't capture any goby. Then starting in summer 2018, we're capturing goby consistently. Go to Utica. Starting in 2019, we are now capturing round goby, albeit at low densities. And we started using electrofishing in 2020 as part of this project, and we captured a single round goby at this location. So turn our attention now to the eDNA, where the story really gets interesting. You can see here at Sylvan Beach, strong positives during every survey. This is the number of PCR reps positive. I'm skipping a whole lot of methods that I don't have time to dive into, but just take it on faith here. 16 is the strongest signature you can have here. That means every single PCR rep was positive. So this makes sense, right? Strong DNA signature related to high catch rates. Going to Rome. This is interesting, right? We have weak positives for 2 surveys here. Then the DNA signature explodes, and that coincides perfectly with when we start catching goby in our trawl. Utica. This one was a head scratcher for us because we had strong DNA positives right from the inception of the project, yet we didn't start catching goby in 2019. And ultimately, the explanation here, in 2018, we discovered about a mile upstream of where we were sampling, there was a high density goby population near Lock 20. The eDNA was able to detect the signature of that population, but since goby weren't present at our immediate study area, the trawl wasn't catching them. Next site, no detections until 2019. Then we have repeated detections using both of our molecular markers. Sure enough, wouldn't you know it, 1 year later, we capture goby here in 2020. The pattern here has been repeated DNA positives tend to translate to captures the following year. So if we use that as the measuring stick going forward, looking at the 2020 eDNA data, we might expect to see captures at a number of these locations where we have repeated eDNA positives in 2020. And of course, as I'm sure many of you have noticed, we even have one hit here in the estuary. Now, that means there was DNA. That doesn't mean there's fish. The two are not synonymous, but obviously it is a warning sign. And if you're eyeing the 2017 data here, I can talk your ear off about what that may or may not mean, but that's a conversation for another day and a longer talk. So other things I want to share on this research. Community impacts. We have a time series of tessellated darter data from our Utica location in which they basically have every single year, if not worse. I spoke to Cornell researchers recently at the American Fisheries Society New York chapter meeting. Cornell studies the heck out of Oneida Lake. They told me prior to round goby invasion, darters were captured in virtually every trawl. In the past 5 years, Cornell has captured 2 darters, none in the past 3. So this suggests that darters are functionally extirpated from Oneida Lake. And it also suggests that what we're seeing in the canal is probably not just noise and is probably real, representing goby impacts. Now, this next slide here, I promise you will not be scientific in any way, but I think it is, Try that one more time. I do think it is informative. This is our office aquarium. We've got a few round goby and a couple rainbow darters and one greenside darter. Watching how they interact really helps you understand the time series and darter data. I mean, the darters are relegated to the poorest habitat in the tank. They have to put themselves physically at risk to acquire any food. This is the dominant male, by the way, goby that just came out. He was guarding a nest in the back of the tank. But, you know, again, in no way scientific, but it is instructive, I think, to actually watch the interactions firsthand. So I hope you enjoy that. I always think that's neat. So where do you go from here? We're going to continue sampling in 2021. We're looking to add 1 or 2 sites in the upper Hudson River, moving towards Lake Champlain. Our normal partner on this project, the Mohawk River Basin Program of the DEC, is really not able to follow us and support us into the upper Hudson. So we are actually looking for new partners to expand our network of sites. And I want to talk a little bit about what goby mean for the Hudson, because I think that's probably what this group is most interested in hearing. First, the obvious question, are goby in the Hudson right now? You know, a year ago, I would have pretty confidently said, I don't think so. I'm going to say a very uneasy maybe right now after the events of 2020. What effects might we see if round goby get into the Hudson and are fairly successful there? And those are big ifs. We might expect to see size structure changes to our zebra mussel populations. After talking to Dave Strayer recently, who was a great help on this, he explained to me that blue crabs have been putting a herding on the larger zebra mussels, meaning much of the zebra mussel population are very small, young of the year mussels. That's like a buffet line for round goby. So goby may come in and put a squeeze on the small end of the zebra mussel size structure. And it'd be really interesting to see what actually happens going forward with the zebra mussel population. If round goby are really successful, and this is a big if, because I don't think that's a foregone conclusion, they could impact zebra mussels enough to maybe reverse some of the impacts zebra mussels have had on this ecosystem. You know, and there's been a lot of research looking at that, and it's time to start thinking about, you know, could we go the other way on a few of these? And on that note, I really want to make the point, are we prepared to monitor these changes? Because it seems to me that goby have the potential to change one of the greatest ecosystem engineers that we have right now. And I understand Carrie has had a tremendous continuous record, both of juvenile and adult draconid densities. And I believe that record is expected to be discontinued this year. And I can't think of a more important data set to extend into the future than our zebra and quagga mussel data. So I would employ all the parties involved to see if we can find a way to continue collecting this data, because this is critical for us to understand any ecosystem level effects from this invasive species. I would also add, we need to think about the contaminant data that we have. Obviously, the Hudson has a long history of contaminated sediments. If goby are successful and eat a lot of draconid mussels that may be high in contaminants, we need to start thinking about contaminant levels in our walleye, our smallmouth bass, our catfish, our bald eagles, our blue herons, right? I mean, the list goes on, but I would pose the open-ended question, do we have that baseline data to try to see if we have major impacts from round goby invasion? And you can't talk about the Hudson without at least mentioning eel. It is very unclear, to me at least, what we should expect from goby-eel interactions. I think eel are going to find goby to be a very easy food source, and eel are so abundant, especially in some of the tribs to the Hudson, they may actually pose an impediment to round goby invasion, or they may limit round goby density. So that's a real interesting dynamic to keep an eye on. So I promised I'd talk about something other than goby, so here goes. Northern snakehead, one of the other invasive fish threats looming on the horizon. Snakehead tend to be a top predator in any ecosystem that they invade. They can breathe air through a lung-like organ, which leads to them getting all sorts of nicknames such as Frankenfish, and stories of snakehead, walking through people's lawn and attacking dogs and children. And of course, these are all ridiculous, but juvenile snakeheads can slither through grass and go in between small bodies of water for a couple days. That has been documented. snakehead really burst onto the scene in New York in 2019. You might remember the story of Rocky the snakehead. This was a aquarium fish. I believe it was actually a giant snakehead, not a northern snakehead, but that was legally obtained. And at some point, DEC became concerned about the threat this posed to ecosystems. But what a headline here, right? A Syracuse man says state environmental conservation officers are going to execute his 10-year-old pet fish today. I mean, what a story, right? I mean, this really elevated the high profile nature of snakeheads as a major household name, at least in the state of New York for sure. And Rocky was not executed, by the way, in case you were worried. He died a natural death a few years later. Flathead catfish, another top predator, at least they are native to North America, that's a plus. Present in most of the major watersheds to the south. This is a top predator that we would expect to compete with invasive channel catfish in the estuary if they are in fact introduced. And my last one, got to talk about Asian carp, obviously. That's most high profile invasive fish, probably in North America right now. Unlike snakehead, which might exert top-down effects on Hudson River ecosystems, this is the opposite. These guys are planktivores. We would expect them to exert bottom-up effects. Asian carp are probably the worst of these fish invaders in terms of they pose safety hazards to boaters. They can transform entire ecosystems based on their effects on plankton populations. At least with goby and snakehead, you can argue there may be some silver lining positive effects. I'm not sure there are any with Asian carp. I mean, this is the worst set of species that we could get introduced into the Hudson in my vantage point. And with that, I need to acknowledge the Mohawk River Basin Program, which has been a tremendous partner funding our round goby research. And I also want to thank Dave Strayer for contributing some of his extensive knowledge to help me put together the talk. And with that, I will stop and hope that we're at least close to being on time.
All right. Well, thank you to our speakers. So we have gotten some questions that came in. So Jim and I are going to tag team this. And I'll just first put out sort of a general question for those of you who rely on modeling in your predictions on what climate will mean for changes. If we, scientific community, are to use modeling to predict changes in the Hudson River, should we emphasize near-term, iterative, sort of the short-term projections that are routinely updated, or put emphasis on longer-term projections? Which would be, which would you think would be more useful and better way to go for understanding potential changes?
This is Stuart. So, as usual, I'm going to be argumentative. We have to choose between the two because you get different things from it. The short term gets you sort of some motivation, have people take things seriously, but many of these changes aren't going to occur to a great extent or be really well documented, well supported for a long time. So can't we have it all?
So both would be the answer. Anybody else have anything to add to that?
I think Kathy, go ahead.
I would jump in on that too and say that, I think it's not only about sort of motivating interest, but I think it really depends on the question and the need for the information. So I think, if you want information on potential risk of pathogens in shellfish, that may be informed by models that take into account present day, more of like a near real-time model that's accounting for current conditions. Whereas if you're planning for fisheries management, maybe you need a framework that will enable you to look 3 to 5 years out. And for long-term planning, these longer range projections. So I really think about it as aligning with the decision-making environment in which you're operating, and that there's a plethora or range of opportunities to plug in with climate or environment or ecosystem informed forecasts or projections at many different scales.
Okay.
Chuck, I'll go,
Since we have you, you
I'll
go
go
ahead, Jim.
I was going to do the same thing you did, I think.
Yeah.
Kathy, if you want to, there's a question from Lewis Kleiman on if you could talk a little bit about the relationship between the ocean warming and Hudson River, at least as far as Poughkeepsie and East River. Like, I guess trying to understand sort of magnitude of changes.
All right, can you hear me?
Yep.
Okay. I don't know that I have, a great answer to that beyond kind of what I showed in one slide that I had that was looking at warming rates from the ocean into the coastal waters and then into the estuary. And so we do see that generally the ocean is warming more rapidly and progressively, the rates are weakening as you move into the estuary. But they are all still positive. So all of these areas are warming. And, we have done a little bit of work looking at projections in rivers, but not in the Hudson. So I don't think I can speak to, sense of what might be coming in the future in terms of how the ocean warming may align with Hudson River warming. But I hope that gives you a bit of insight from the past observations that we do have to turn to.
Great, thanks.
I'll move on to Chris Neck. I have a question for you. At some point in your presentation, you're talking about intakes and like no more. Can you explain what you meant by, I believe you said there'd be no more intakes as of 2017. That came from Veronica Thompson.
What I meant was from large industrial factories such as cement plants and specifically the power plant, kind of the big companies that really paid for the monitoring program along the Hudson River. Obviously, there's large numbers of intakes along the river, mostly smaller ones, but yeah.
Right. So your focus was more that the monitoring program that was connected with the power plant's use of Hudson River,
Right.
Since
right.
they pretty much either no longer withdraw water or they've put in technologies to protect fish, they're no longer conducting the monitoring that had gone on for many, many years. Is that
Right.
correct? Yeah, okay.
Thank you. Yeah, I think I misspoke. I'm sorry.
I have one from Stuart, Werner Mueller. He's questioning where the 40% increase in freshwater inflows come from, commenting that seems very high.
So first, I agree, it does seem very high. All the flow data that we use come from USGS, and it's it's summed up. It's a green island. Plus, we often add in for the tributaries that are gauged. So USGS water data are pretty good, very good. And so that's ultimately the source. And so it's an annual mean, it's 40% increase over the last about quarter century. We are getting more precipitation and more intense precipitation. So I think it does make some sense. It's big, but I believe it's true.
Thanks.
Sure. And I guess, Stuart, a related question. So this increase in intensity and frequency of storms, how do you think that's going to affect Hudson River aquatic habitats?
The short answer is not well. And, high flow is sort of bad for many things. The only thing it's good for is for a river that moves out onto its floodplain and, can deposit sediment and help soil fertility and things like that. That's not really what's happening in the Hudson. These high flow events probably deliver too much sediment at the wrong time. And I do think there's sort of a right amount of sediment. But it has a bunch of effects both on the creatures and organisms in the river as well as our infrastructure. And I think one of the biggest effects is the human response. You know, if things look a little bit dangerous, the human response is let's build something big to keep us from danger. And that's not necessarily good for the river in those cases.
So I have one for Dave Strayer. Carolyn Hall is wondering if you mentioned that there was a parasite that could be the cause of the electrification of rainbow smelt rather than, as you pointed out, the their temperature data. She's wondering if it could be a combination of the two.
Yeah, sure could be. I'm not a fish veterinarian, but quite a few of these parasites are more likely to infect fish and more likely to kill fish if the fish is stressed. So you could imagine a case where the smelt are a little bit stressed by the high temperature, and maybe this parasite that doesn't cause them a lot of trouble in a cooler year might cause them more trouble in a warm year.
Thanks.
All right, and then I've got a question for Scott George. So what are the prospects for blue catfish invasion in the Hudson River?
Yeah, it's a great question. And I would say, as far as I know, I would say comparable to that of the flathead cat. And, that potentially could have been woven into to that slide as well. My understanding actually is that they were each intentionally introduced to some watersheds in Virginia, and obviously the northward expansion is evident. But yeah, I would say comparable to that of the flathead is the short answer.
So I have one from Bonnie Harkin. She's wondering, how do you anticipate the projected increase in the intensity and frequency of storms will affect Hudson River habitats? It might have been directed at Stuart, but I think anyone can take it.
I think Chuck just paraphrased that, and my short answer would be most of the effects are probably not very good. And probably the biggest one is this human response by building some sort of protective structure.
I'm sorry, I missed that. Yes, sorry. Thanks.
Chuck's out in front as usual.
I was too busy reading to listen to your response.
You guys are doing fine. You're doing fine.
So I have a couple on sort of human potential impacts to human use of the river. One is from Lois Kay at Hudson River swim events. There's often jellyfish stinging folks. Do we anticipate that jellyfish are going to continue to increase in the Hudson with warming? That could be for anyone who might know.
I will just chime in to say that, I think further up north, where I work mostly in the Gulf of Maine, we have certainly been getting increasing reports of jellyfish blooms and jellyfish sightings or unusual jellyfish. I would say particularly during the heat wave in 2012. So there's some indication that perhaps that's related to warming, but as someone pointed out, a lot of the data that I lean on in my research is really, long-term monitoring data. And we just, don't have that type of long-term record for jellyfish. So it's a lot more variable. The observation record is more gappy. So it's harder to really put those relationships together.
I have one more here. We've been mostly focused on ecological impacts, but again, looking at sort of a human effect or human health issue. Does anyone have any information or any idea about relationship of an increase in pathogenic infections to humans as a function of the rise in river water temperatures?
Well, I guess it's an obvious point, but we can expect human pathogens that are more common in warmer areas to start appearing more frequently in the Hudson. So things that either might be in the Carolinas all the way down to Florida might turn up in the Hudson as the Hudson warms, or things might come from some distant part of the world, just as I've said for fish diseases.
I mean,
Right.
the other one is are the blue-green blooms, which both heat waves and generally warmer temperature. There's a good record from Sarah Fernald on the effect of water temperature. It's not directly a human pathogen, but I sure don't want them.
Right. Okay. Were there any others there, Jim, that we didn't cover?
I don't see, I think we got them all. So just I wanted to thank everyone again, all our speakers, excellent job. And thanks everyone for participating. Oh,
Yeah,
Chuck,
thank you very much.
You want to tell us about the next two seminars?
Sure. So part 4 will be on March 15th from 1:30 to 3:30. And there we'll be looking at the science of harvest. So basically what we hope to cover there is how resource managers are dealing with managing fisheries in the face of a changing climate. And again, what kind of stress that places on fish populations. And then finally, on March 22nd, we kind of pull it all together. Some of the speakers that you've heard already will come back and kind of give a big picture thinking kind of brief presentation and have open discussion with all the panelists at that point. So I hope that you can participate on this. And if you have to miss any of these sessions, they are available on Hudson River Foundation's website. They're all being recorded.
Yep, this should be up
Thank you.
tomorrow. Thanks, guys. Thank you, everyone.